Welcome back to Biology A. It's Monday, it's a lab day, and we're going to be using today's time to identify biological compounds in different samples. Continuing on with this week's big idea, this unit's big idea, to use evidence to explain how carbon, hydrogen, oxygen, and other specific elements form the four major organic molecules. We've got 3 lesson targets for today. We're going to explain the function of indicator solutions. There's actually not a fancier name for that. They are solutions that are water-based, and they indicate when a substance is present, okay? That's really all they do. Then we're going to describe an experimental process to use those indicators to identify the presence of specific molecules, and we're going to practice our scientific method by gathering and analyzing data and forming a conclusion. All right, carbon and macromolecules. We've got 4 that we've been talking about. We are going to be testing for 3 of them today. We've discussed carbohydrates, lipids, and proteins so far. We have not discussed nucleic acids yet. That's okay, so we're not going to be testing for those in these samples. You're going to add, again, these indicators to common samples that you might find in your kitchen. Oil, sugar water, egg white, which is another word for albumin. That's what we're going to be using for proteins. And then, depending on what color change we see, qualitative observations, then that's going to indicate or not whether or not the substance we're testing for is there. All right, some basic steps to complete the lab. You're going to have to download 2 documents from your content. They are the pre-lab and tutorial and the lab report that accompanies it. There are videos that will accompany both of these, and I'm going to have you do the virtual lab in class today. You're going to have to complete the lab report on your own. I'm not giving you all the answers. You're going to submit the report by tonight at 11:30. If you need some extra time, email me. But I'm going to be going through the process with you in these videos, so you should be able to reference those. If I wanted to give you extra time, but I really want you to have that overdue notice just to help you remember to turn it in, because I still have people that haven't turned in the first one. Okay, so the 2 documents you're going to need, if you go into your content, you'll find them in 2B Laboratory, Investigating Biological Compounds module. They're the first 2 things that should pop up for you. Download them and open them. The pre-lab, I have taken all of the reading for the content. From last year, you would have had to follow different links, but I compiled it all for you. This is what I call background reading. Here's the main idea and the lesson targets still there for you in case you want to review those. Here's a little graphic organizer to help you visualize things. But this part here is where it talks about what a chemical indicator is and what the role it plays basically in this chemistry. And then this is where it talks about the specific indicators we're going to use. Don't neglect these readings, or I should say this 1 page of reading, because there are going to be some answers to the questions that come up in here. And you're going to say, oh, Mrs. McAnally, where do you find the answer to blah, blah question? And I'm going to say, it's right there in the reading. It's called a right there question, all right? And I'm not being sarcastic. I'm telling you to go back and look in the reading. In the reading, you should see that the whole point, again, of this lab is we're using indicators because if you take, let me turn on my camera here for a minute. Okay, if you take a test tube and it's got a sample in it, like I said, there's going to be sugar water, oil, and egg whites, which is another word for protein, right? It's albumin, I should say, is another word for it's what we're using for protein. If you take an indicator and you're going to put each indicator into all of those substances, and if you drop it in there and it goes from clear or white to a different color, that's indicating with a qualitative observation that something has changed. And the something that has changed is that the indicator you put in there has found the specific molecule that it's looking for, either fat, lipid, or protein. Oops, those 2 are the same. Lipid, carb, or protein. And it flags for you visually, yep, it's there, or no, it's not. So if you don't see a color change, there is no chemical reaction that you need to notice because there's no reaction between the indicator and the specific molecule. On the second page of the tutorial, you'll find a little blurb about safety, but I think we all understand this is virtual. Okay, so keep safety in mind when you do things at home as a demo or with me. You're going to find on page 2 where you begin the procedural steps. So here is where it talks about opening the link, download the lab report. It's not the 2.8 lab report anymore. I apologize. I should fix that. It will be fixed in your sheet. And then it's not going to, I've put this graphic here just so that when we talk about this in the pre-lab, which I think I did on Friday for you guys, I've described everything here. Here is the vegetable oil, here's the water, here's the milk. And then you're going to see a starch solution, which just means sugar water, iodine, those are the 2 indicators. That's Sudan 3. Anyway, these are all my eyes are too old to see that tiny stuff, but you can read it. These are on the top shelf above where you're going to be working in the lab. So part 1 is the tutorial for the starch portion of the test. Okay, so you're in the tutorial, you're actually doing the first macromolecule test, which is starches. So you need to follow these steps to find out whether or not that sample is a starch or has starch in it. So where I've got these blue words, that's where I want you to, that's what the question is. The point of this part is this question, and then it tells you answer this question on your lab write-up. And if you look at your lab write-up, you'll see part 1, there's a place for you to insert your question, or I should say insert your answer. Part 2 is where it's addressing the protein test portion of the lab. Third page, you'll find part 3, part 4, and part 5. So part 3 wraps it up with the lipid portion of the testing. Part 4 is where you're testing the unknown. I'm not going to tell you what it is. You've got to tell me what the unknown solution has in it. 5 is another sort of an unknown. 5 is milk. So if you thought really hard about what might be in milk, or if you went and read the label on the milk, you could probably assume that you could figure that out. I want you to assume this is vitamin D or full fat milk or whole milk. That's the steps for that. I'm going to talk about this part next. All right, so the last part is where you're actually going to be using your scientific method skills. So you're going to identify parts of the scientific method. You're going to identify what the control is. You're going to talk about how you can verify that your data is accurate because you know what you're talking about, because you know what an indicator is. So this is where you describe what an indicator is and how it functions. And then the last part, I want you to notice 6 points. It's the largest ticket item on this whole report. It needs to be a paragraph, which is more than 3 sentences. It needs to be complete sentences. Feel free to spell check. Use your grammar spell check functions. I mean, I'm not asking you to turn in a rough draft. I want a final conclusion paragraph. And it needs to have evidence from your data table to support, evidence from the background reading to support, etc. Show me what you can do, all right? There will be a chance. I'll give you feedback. You can have a chance to resubmit, but do your best the first time. All right, in order to do the virtual lab, you're going to have to, you can go stay in the same content. There's a link right there, it says. When you do the virtual lab, you are going to have to enable Adobe Flash Player. Okay, don't be afraid. You're not going to get a virus. You're going to be fine. You need to make this work in order to access the lab. Don't come to me and say, all I see is a white screen, Mrs. McAnally, because I don't know what's going on. My lab is broken. It's not broken. You need to enable Flash Player. Now, it might show up somewhere here as a little puzzle piece in your menu. I should say your browser menu or your search bar, or it might just be a big white space with a thing that says, hey, got to enable Flash Player. So you have to do that or it will not work. Okay. You're going to start with the tutorial when you get to this page here. It's going to ask you, do you want to open it as an external resource? Yes. Then you're going to go to start with the tutorial portion. I will do a little video for you, and then do the lab. That's parts 2 through 5. All right, finally, we're talking about the lab report itself. What do you have to turn in? This is the sheet you have to turn in. The tutorial is for you to read through on your own, mark up how you want, take all the notes you want. The lab report is what you turn into me. Don't do your work on the tutorial, please. All right, so download this and open it. Where it's got blue ink here, that's where it's telling you to replace this text with your answer. That's where I want you to write your answer, okay? Also, if you look at the data table, I've already X'd out boxes that do not need to be tested, all right? So don't worry about testing those, or test them and see what happens, but there shouldn't be any reactions there. So again, the data table, no test required. You just need to look at what's happening with these. Go ahead and fill them in as best as you can. What does no change mean? I've already answered that one for you earlier in this video. Go back and review if you need to. The other thing you need to notice on the lab report is that I've put the points for each question. It's not an overall, you just get it a good grade because I like you. I have to give you grades, your grade based on how you've answered each of these questions. Don't forget that the last question, this says 8 points. 8, 10, 13, 14, 15, 16, 17, 18, 19. Oh, plus the day. Yeah, plus the lab report. Whatever the points are, that's what you get, okay? Okay, just to make sure we're on the same page, we are hitting these 3 learning targets today. Make sure that you're ready to do it, and then at some point you need to be able to explain the function of indicator solutions. You'll do that on your lab report. Describe an experimental process. You'll probably do that on your test. And then practice the scientific methods of gathering and analyzing data. That's the part you'll prove to me when you turn in your lab report. So make sure you can do these and that you're comfortable. And if you are somewhere at C or D, you need to come and seek me out. Okay, so that was the video for the instructions and walkthrough for the identifying biological compounds lab we're doing today. And if you have any questions, feel free to ask me in class or email or text me.